Iodine in biology. Iodine is an essential trace element in biological systems. It has the distinction of being the heaviest element commonly needed by living organisms as well as the second heaviest known to be used by any form of life. Only tungsten, a component of a few bacterial enzymes, has a higher atomic number and atomic weight. It is a component of biochemical pathways in organisms from all biological kingdoms, suggesting its fundamental significance throughout the evolutionary history of life. Iodine is critical to the proper functioning of the vertebrate endocrine system and plays smaller roles in numerous other organs, including those of the digestive and reproductive systems. An adequate intake of iodine-containing compounds is important at all stages of development, especially during the fetal and neonatal periods, and diets deficient in iodine can present serious consequences for growth and metabolism. Functions. Thyroid. In vertebrate biology, iodine's primary function is as a constituent of the thyroid hormones, thyroxine T4 and triiodothyronine T3. These molecules are made from addition condensation products of the amino acid tyrosine and are stored prior to release in an iodine-containing protein called thyroglobulin. T4 and T3 contain 4 and 3 atoms of iodine per molecule. Respectively, iodine accounts for 65% of the molecular weight of T4 and 59% of T3. The thyroid gland actively absorbs iodine from the blood to produce and release these hormones into the blood, actions which are regulated by a second hormone called thyroid-stimulating hormone TSH, which is produced by the pituitary gland. Thyroid hormones are phylogenetically very old molecules which are synthesized by most multicellular organisms and which even have some effect on unicellular organisms. Thyroid hormones play a fundamental role in biology, acting upon gene transcription mechanisms to regulate the basal metabolic rate. T3 acts on small intestine cells and adipocytes to increase carbohydrate absorption and fatty acid release, respectively. A deficiency of thyroid hormones can reduce basal metabolic rate up to 50%, while an excessive production of thyroid hormones can increase the basal metabolic rate by 100%. T4 acts largely as a precursor to T3, which is, with minor exceptions, the biologically active hormone. Via the thyroid hormones, iodine has a nutritional relationship with selenium. A family of selenium-dependent enzymes called deiodinases converts T4 to T3, the active hormone, by removing an iodine atom from the outer tyrosine ring. These enzymes also convert T4 to reverse T3 or T3 by removing an inner ring iodine atom, and also convert T3 to 33-deodothioronine T2 by removing an inner ring atom. Both of the latter products are inactivated hormones which have essentially no biological effects and are quickly prepared for disposal. A family of non-selenium-dependent enzymes then further deiodonates the products of these reactions. In truth, the total amount of iodine in the human body is still controversial, and in 2001 M.T. Hayes published in Thyroid that it is surprising that the total iodine content of the human body remains uncertain after many years of interest in iodine metabolism. Only the iodine content of the thyroid gland has been measured accurately by fluorescent scanning, and it is now well estimate of 515 mg in the normal human thyroid. But similar methods are not available for other tissues and for the extrathyroidal organs. Many researchers reported different numbers of 10-50 mg of the total iodine content in human body. Selenium also plays a very important role in the production of glutathione, the body's most powerful antioxidant. During the production of the thyroid hormones, hydrogen peroxide is produced in large quantities, and therefore high iodine in the absence of selenium can destroy the thyroid gland, often described as a sore throat feeling. The peroxides are neutralized through the production of glutathione from selenium. In turn, an excess of selenium increases demand for iodine, and deficiency will result when a diet is high in selenium and low in iodine. Extrathyroidal iodine. Extrathyroidal iodine exists in several other organs, including the mammary glands, eyes, gastric mucosa, cervix, cerebrospinal fluid, arterial walls, ovary, and salivary glands. In the cells of these tissues, the iodide ion enters directly by the sodium iodide symporter NIS. Different tissue responses for iodine and iodide occur in the mammary glands and the thyroid gland of rats. The role of iodine in mammary tissue is related to fetal and neonatal development, but its role in the other tissues is not well known. It has been shown to act as an antioxidant and antiproliferant in various tissues that can uptake iodine. Molecular iodine has been shown to have a suppressive effect on benign and cancerous neoplasias. The U.S. Food and Nutrition Board and Institute of Medicine recommended daily allowance of iodine ranges from 150 micrograms per day for adult humans to 290 micrograms per day for a lactating mother. However, the thyroid gland needs no more than 70 micrograms per day. The higher recommended daily allowance levels of iodine seem necessary for optimal function of a number of other body systems, including lactating breasts, gastric mucosa, salivary glands, oral mucosa, arterial walls, thymus, epidermis, choroid plexus, and cerebrospinal fluid, among others. Other functions. Iodine and thyroxine have also been shown to stimulate the spectacular apoptosis of the cells of the larval gills, tail, and fins during metamorphosis in amphibians, as well as the transformation of their nervous system from that of the aquatic, herbivorous tadpole into that of the terrestrial, carnivorous adult. The frog species Xenopus laevis has proven to be an ideal model organism for experimental study of the mechanisms of apoptosis and the role of iodine in developmental biology. Agents containing iodine can exert a differential effect upon different species in an agricultural system. The growth of all strains of Fusarium verticilliads is significantly inhibited by an iodine-containing fungostatic J1629-34 EC at concentrations that do not harm the crop. This might be a less toxic antifungal agricultural treatment due to its relatively natural chemistry. Dietary recommendations. The U.S. Institute of Medicine, IOM updated estimated average requirements, EAR, and recommended dietary allowances RDA for iodine in 2000. For people age 14 and up, the iodine RDA is 150 mg/day, the RDA for pregnant women is 220 mg/day, and the RDA during lactation is 290 mg/day. For children 1-8 years, the RDA is 90 mg/day. For children 8-13 years, 130 mg/day. As a safety consideration, the IOM sets tolerable upper intake levels UL for vitamins and minerals when evidence is sufficient. The UL for iodine for adults is 1100 mg/day. This UL was assessed by analyzing the effect of supplementation on thyroid-stimulating hormone. Collectively, the EAR RDAs, AA and UL are referred to as dietary reference intakes DRI. The European Food Safety Authority EFSA refers to the collected set of information as dietary reference values with population reference intake PRI instead of RDA and average requirement instead of EAR. AI and UL are defined the same as in the United States. For women and men ages 18 and older, the PRI for iodine is set at 150 mg/day. The PRI during pregnancy or lactation is 200 mg/day. For children ages 1-17 years, the PRI increases with age from 90 to 130 mg/day. These PRI are comparable to the U.S. RDA with the exception of that for lactation. The EFSA reviewed the same safety question and set its adult UL at 600 mg/day, which is a bit more than half the U.S. value. Notably, Japan reduced its adult iodine UL from 3,000 to 2,200 UG/day in 2010, but then increased it back to 3,000 UG/day in 2015. As of 2000, The median observed intake of iodine from food in the United States was 240 to 300 mg/day for men and 190 to 210 mg/day for women. In Japan, consumption is much higher due to the frequent consumption of seaweed or kombu kelp. The average daily intake in Japan ranges from 1,000 to 3,000 mg/day. Previous estimates suggested an average intake as high as 13,000 mg/day. Labeling. For U.S. food and dietary supplement labeling purposes, the amount in a serving is expressed as a percent of daily value %DV. For iodine specifically, 100% of the daily value is considered 150 mg, and this figure remained at 150 mg in the May 27, 2016 revision. A table of the old and new adult daily values is provided at reference daily intake. Food sources. Natural sources of iodine include many marine organisms, such as kelp and certain seafood products, as well as plants grown on iodine-rich soil. Iodized salt is fortified with iodine. According to a Food Fortification Initiative 2016 report, 130 countries have mandatory iodine fortification of salt and an additional 10 have voluntary fortification. Deficiency. Worldwide, iodine deficiency affects 2 billion people and is the leading preventable cause of mental retardation. Mental disability is a result which occurs primarily when babies or small children are rendered hypothyroidic by a lack of dietary iodine. New hypothyroidism in adults may cause temporary mental slowing, but not permanent damage. In areas where there is little iodine in the diet, typically remote inland areas and semi-arid equatorial climates where no marine foods are eaten, iodine deficiency also gives rise to hypothyroidism, the most serious symptoms of which are epidemic goiter, swelling of the thyroid gland, extreme fatigue, mental slowing, depression, weight gain. The addition of iodine to table salt, so-called iodized salt, has largely eliminated the most severe consequences of iodine deficiency in wealthier nations, but deficiency remains a serious public health problem in the developing world. Iodine deficiency is also a problem in certain areas of Europe. In Germany, an estimated $1 billion in healthcare costs is spent each year in combating and treating iodine deficiency. Iodine and cancer risk. Source: breast cancer. The mammary gland actively concentrates iodine into milk for the benefit of the developing infant and may develop a goiter-like hyperplasia, sometimes manifesting as fibrocystic breast disease, when iodine level is low. Studies indicate that iodine deficiency, either dietary or pharmacologic, can lead to breast atypia and increased incidence of malignancy in animal models, while iodine treatment can reverse dysplasia, with elemental iodine having been found to be more effective in reducing ductal hyperplasias and perilobular fibrosis in iodine-deficient. On the observation that Japanese women who consume iodine-rich seaweed have a relatively low rate of breast cancer, iodine is suggested as a protection against breast cancer. Iodine is known to induce apoptosis in breast cancer cells. Laboratory evidence has demonstrated an effect of iodine on breast cancer that is in part independent of thyroid function, with iodine inhibiting cancer through modulation of the estrogen pathway. Gene array profiling of the estrogen-responsive breast cancer cell line shows that the combination of iodine and iodide alters gene expression and inhibits the estrogen response through upregulating proteins involved. Whether iodine/iodide will be useful as an adjuvant therapy in the pharmacologic manipulation of the estrogen pathway in women with breast cancer has not been determined clinically. Gastric cancer. Some researchers have found an epidemiologic correlation between iodine deficiency, iodine-deficient goiter, and gastric cancer, a decrease in the death incidence from stomach cancer after iodine prophylaxis. In the proposed mechanism, the iodide ion functions in gastric mucosa as an antioxidant, reducing species that detoxifies poisonous reactive oxygen species, such as hydrogen peroxide. Precautions and toxicity. Elemental iodine. Elemental iodine is an oxidizing irritant, and direct contact with skin can cause lesions, so iodine crystals should be handled with care. Solutions with high elemental iodine concentration, such as tincture of iodine, are capable of causing tissue damage if use for cleaning and antisepsis is prolonged. Although elemental iodine is used in the formulation of Lugol's solution, a common medical disinfectant, it becomes triiodide upon reacting with the potassium iodide used in the solution and is therefore non-toxic. Only a small amount of elemental iodine will dissolve in water, and adding potassium iodide allows a much larger amount of elemental iodine to dissolve through the reaction of It2O3. This allows Lugol's iodine to be produced in strengths varying from 2% to 15% iodine. Elemental iodine is poisonous if taken orally in large amounts. 2–3 grams is a lethal dose for an adult human. Potassium iodide, on the other hand, has a median lethal dose LD50 that is relatively high. In several other animals, in rabbits, it is 10 g/kg. In rats, 14 g/kg, and in mice, 22 g/kg. The tolerable upper intake level for iodine, as established by the Food and Nutrition Board, is 1100 ug/day for adults. The safe upper limit of consumption set by the Ministry of Health, Labor and Welfare in Japan is 3000 ug/day. The biological half-life of iodine differs between the various organs of the body, from 100 days in the thyroid to 14 days in the kidneys and spleen to 7 days in the reproductive organs. Typically, the daily urinary elimination rate ranges from 100 to 200 ug/L in humans. However, the Japanese diet, high in iodine-rich kelp, contains 1000 to 3000 ug of iodine per day, and research indicates the body can readily eliminate excess iodine that is not needed for thyroid hormone production. The literature reports as much as 30,000 ug/L, 30 mg/L of iodine being safely excreted in the urine in a single day, with levels returning to the standard range in a couple of days, depending on seaweed intake. One study concluded the range of total body iodine content in males was 12.1 mg to 25.3 mg, with a mean of 14.6 mg. It is presumed that once thyroid-stimulating hormone is suppressed, the body simply eliminates excess iodine, and as a result, long-term supplementation with high doses of iodine has no additional effect once the body is replete with enough iodine. It is unknown if the thyroid gland is the rate-limiting factor in generating thyroid hormone from iodine and tyrosine, but assuming it is not, a short-term loading dose of 1 or 2 weeks at the tolerable upper intake level may quickly restore thyroid function in iodine-deficient patients. Iodine vapor is very irritating to the eye, to mucous membranes, and in the respiratory tract. Concentration of iodine in the air should not exceed. When mixed with ammonia and water, elemental iodine forms nitrogen triiodide, which is extremely shock sensitive and can explode unexpectedly. Iodide ion. Excessive iodine intake presents symptoms similar to those of iodine deficiency. Commonly encountered symptoms are abnormal growth of the thyroid gland and disorders in functioning, as well as in growth of the organism as a whole. Iodide toxicity is similar to but not the same as toxicity to ions of the other halogens, such as bromides or fluorides. Excess bromine and fluorine can prevent successful iodine uptake, storage, and use in organisms, as both elements can selectively replace iodine biochemically. Excess iodine may also be more cytotoxic in combination with selenium deficiency. Iodine supplementation in selenium-deficient populations is theoretically problematic, partly for this reason. Selenoscistine, abbreviated as SEC or U in older publications also as C-size, is the 21st proteinogenic amino acid, and is the root of iodide ion toxicity when there is a simultaneous insufficiency of biologically available selenium. Selenoscistine exists naturally in all kingdoms of life as a building block of selenoproteins. Hypersensitivity reactions. Some people develop a hypersensitivity to compounds of iodine, but there are no known cases of people being directly allergic to elemental iodine itself. Notable sensitivity reactions that have been observed in humans include: the application of tincture of iodine may cause a rash. Some cases of reaction to povidone iodine betadine have been documented to be a chemical burn. Medical use of iodine compounds, i.e., as a contrast agent, can cause anaphylactic shock in highly sensitive patients, presumably due to sensitivity to the chemical carrier. Cases of sensitivity to iodine compounds should not be formally classified as iodine allergies, as this perpetuates the erroneous belief that it is the iodine to which patients react, rather than to the specific allergen. Sensitivity to iodine-containing compounds is rare, but has a considerable effect given the extremely widespread use of iodine-based contrast media. However, the only adverse effect of contrast material that can convincingly be ascribed to free iodide is iodide mumps and other manifestations of iodism.